Welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines. People just like you working to understand viruses and how they affect you. During the COVID-19 pandemic, we are talking with researchers involved in coronavirus and COVID-19 related research so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On April 21st, 2021, we talked with Dr. Ellebedy, an immunologist who studies humoral immune responses after virus infection. Ali obtained his BS in pharmaceutical sciences from Cairo University, Egypt, and then his PhD studying influenza virus with Richard Webby at St. Jude Children's Research Hospital in Memphis, Tennessee. He then moved to Emory University in Atlanta, where he was a postdoctoral fellow in the laboratory of Rafi Ahmed. At Emory, Ali studied human B-cell responses to influenza and Ebola viruses. In 2017, Ali joined the Department of Pathology and Immunology at Washington University School of Medicine, where his lab continues to study B-cell responses to influenza virus and now SARS-CoV-2. In 2021, he was promoted to an associate professor. Happy to have you with us today. Why don't you tell us about yourself? How did you become interested in virology and immunology?
Well, thanks for having me. This actually goes back to when I was an undergrad in pharmacy school in Egypt, in Cairo University. This was, it's specifically, actually, I was in my third year at the time. It was probably 2001, 2002. And at the time we had, it actually was a pharmacy school, so there wasn't much going on in terms of teaching the microbiology, immunology. We had one course in microbiology, and then there was another one about immunology. Actually, immunology didn't have a course by itself. It was part of like a microbial pathogenesis and immunology. And at the time, I remember that was the first lecture that we had actually about immunology. I was like, I don't know what would be the expression for it. I was like, done. This is it. And that's I was fascinating from the first moment. And I started like, you know, and it's really, when you go to a pharm school, especially in Cairo and probably, it's a lot of material to cover. Lots of organic chemistry, lots of inorganic chemistry, lots of all kinds of chemistry, pharmaceuticals, formulation, and all of that, and pharmaceutics, oh my God. And it's a lot of material. So you barely have time to actually do anything outside of your course material. But because of just from the first moment, I was struck. So I was just going to the library and reading, getting the textbook from immunology textbook and starting to read more and more about outside of really of that small piece in the course and just tried. And that's from that moment, it's one of the few moments in actually that I really had a clear that this is it. I'm not even going anywhere else. I'm going to continue on this. And that was the time. And I continued for that. Even after I finished the pharmacy school, so in the way it works in Egypt is, or at least in that education system, that you have actually, you can be, once you graduate and you have a relatively high GPA, kind of, you are offered to be an assistant, teaching assistant in the same school. And this is usually offered for the top, maybe top 10, 20 students. And then they allow you to pick, depends on your rank, is which department you would like to. Again, everyone in my class knew that from that moment that I will be going for this immunology because I want to teach immunology. And so again, everything I decided after that moment was based on this. I want to continue to study immunology. And then when I applied for grad schools in the US, this is the same thing in 2006 when I came. I only got accepted in the University of Tennessee. There are many reasons, but basically we didn't really know how to apply for grad schools there. It's just it was something. Yeah, as an international student, you really don't. For example, one of the things that I remember very clearly is personal statement or a statement of this thing is just, I didn't know what a personal statement is. So we never actually took. I can imagine how my application looked like, and I don't blame 15, 16, I think 15 schools that rejected my application because it was clear to them that I didn't really spend time writing my personal statement. So I came here and then I worked in St. Jude at hostel, which I didn't know existed before I come. But it was I'm very happy that I actually went to Memphis, Tennessee, because of St. Jude is a wonderful place to work on. And from there again, I focused in immunology. It was an interdisciplinary program, so you can pick the field and the track that you want to go for. And again, was very clear that I will go for immunology. The only twist there is that I couldn't find all of the immunology labs. At least at the time at St. Jude, did not have a place for rotation, and I was very disappointed. But one way I did it is I just went for a virology lab. At the time, it was Peter Doherty, who is a Nobel laureate for 1996 Nobel laureate for immunology, who had a part-time. He was partially running his lab in immunology in St. Jude at the time. And he was using influenza virus to study T-cell responses and using influenza as a model. So I remember clearly emailed him multiple times to try to rotate in his lab. And he would say that, I'm spending a lot of time in Australia. And it was a time that it wasn't clear that, and he didn't have interest in actually getting any students. But because he's working on influenza, I thought if I work in an influenza lab, then I can potentially actually do some sort of collaboration with his lab, and that way I can get into the immunology part of things. And this is how it works. So I worked out and ended up working in the Richard Webby lab in and it's pure virology lab, looking at influenza viruses. But from there, from the first moment, my project was really directed. Can we start a collaboration right away? Because that's what I wanted to do. And it worked out well. It's funny because we still, the lab after Peter retired, his main postdoc at the time is now running the lab, Paul Thomas. And funny enough, I still work with Paul. Even at the time, I did the rotation with him, but and then my PhD project collaboration with him, and we, he continued there, but I went different ways, and here I am. I'm still working on influenza actually. So that was really the model that I started. I really wasn't very keen about influenza or any other model, infectious model. I was really keen about studying the immune response to an infectious pathogen. It happened to be influenza because of Peter Doherty there and his lab, but then I really fell in love with influenza, so I'm fascinated by it too. So now I'm working on influenza.
Right. So I guess to follow up on that, can you tell us a little bit about some of the research you were doing before the COVID pandemic to kind of give us sort of a background of the type of questions that you're interested in or what you were thinking about?
So yeah, so what happened after I finished my PhD now is I had a chance to actually now to go to an immunology lab. And that's when I went to Rafi Ahmed's lab in Emory. And I was very, I was very determined. Now I really need to get proper immunology training, and you get that in Rafi's lab. So you go there but then at the time, because of my work in PhD, I developed a huge interest in B cells, and one of the things I told Rafi, and he's really a world. One of the leaders in T-cell immunology. So I told him I'm really interested in B cells, and Rafi was looking for someone to do B cells and influenza in his lab. So for him, it was, of course, yeah. He didn't just do B cells and you do influenza, and I said great. So the other thing that I didn't know before going to his lab is working with humans. I was only looking at animal models. I worked with ferrets and mice during my PhD. And I thought I'll continue to do at least mouse work in Rafi's lab, but he actually was more interested in me working in human. Wasn't very interested in that in the first year, but slowly realized that there are some interesting questions you can address there, and important ones. Um, the one thing I did a lot of work in humans. The one thing that during my time with Rafi, it was only looking in peripheral blood, and that was something I always was not very satisfied with. That although we can answer kind of some important questions, I always thought that this is something not we are not looking at the full picture, and that has been really the motivation for me to what else can we dig deeper, literally. So can we actually look at can we have a sample? Can we do something to allow us to learn more about why this vaccine is not working optimally? And I couldn't do obviously as a postdoc. There's a limit for how much push you can do or how far you can go, and I didn't talk to at that time during the end of or toward the end of my postdoc fellowship, 2015, 16. Emerging studies starting to come about using fine needle aspiration in non-human primates to look at B-cell responses in the draining lymph nodes. And again, in non-human primates, you are allowed to it's a much easier, more feasible studies to do this, not only to look at peripheral blood, but look at B cells as they become activated and differentiate to what we know as germinal center B cells in the draining lymph node. So there were was pioneered by Shane Crotty in La Jolla, and he was working with actually folks at Emory at the time with the non-human primate facility. So I thought this will be something that we can hopefully do in humans at one day and during the time. So when actually I came here at WashU in 2017, I actually during the time I was interviewing, I actually mentioned that this will be something I would like to do, explore this possibility. And I said, I think we just need a radiologist. I was very, at the time, obviously, and still in many aspects, very naive to how things are. But I just talked to Rachel Presti, which is in infectious disease here, and I told her, Rachel, if there's a chance that I can talk to a radiologist, especially in ultrasound, I have this crazy idea about if we can do this monkey studies in human. And she said, well, I do have a friend, and she told me Sherry Tiffy here in radiology she knows her, and she said I can introduce you when you come. So I thought, okay, that's great. So I thought we will just there's a chance, there's a potential. So when I came for probably my first week here, I actually met with Sherry and Rachel, and I said, here's what I would like to do is to recruit healthy people, somehow convince them to give us not only blood, but also allow us to use ultrasound to sample the draining lymph node by fine needle aspiration before and after vaccination. And then I remember that Sherry would tell, well, let's assume you convince them. She asked me many important questions that I wasn't really. She said, let's assume you convince someone to do that. So what are you going to do with the cells? And second, she said, yeah. So that was the first one. And she said, and the second thing is which lymph node you're talking about. And will tell you exactly how very almost clueless I was at the time, because I told her the axillary lymph node, of course, because we are draining axillary. She told me there's probably 30 lymph nodes in that area. Which one you want to sample? I said 30. I thought mice they only have one lymph node in each spot. Why do we have 30 in humans? And that took us a really I would say like a journey to try to decide which one are the right lymph nodes, the right one that should be draining the upper arm area where we get our vaccine. And we eventually managed to find this one after we basically spent the whole first year with some really generous participants, and we figure out which one is most likely is the draining one and how to locate it. And that allowed us now to answer the really an important question, and I'm very excited about that. Does actually influenza vaccination induce a germinal center response in humans? And we have always been, I've looked so much in blood, and I was very, very excited about that we started to open that door. I have to say that at the time, actually, I did call, when I first started, I did call Shane Crotty actually by phone. I told him, Shane, I was very fascinated by your NHP studies, and I want to do this in humans. And he was very encouraging, and he said, yeah, I'm sure that this will be doable, and he gave us some tips, and we took it from there. And these studies have continued all the way for the first 2 years till we got our till of course the pandemic hit us. But we had our first paper published it published during the pandemic. It was actually submitted. That first paper describing our approach and the first results were submitted on March 2nd, 2020. So it was actually a week before the things we start to get the hint that this is going to be a shutdown. And that's when I submitted the paper. We did not realize that this is going to be going that far. I always, and this is a true story, and many people here, you know that I've always resisted because I was very excited at the time with our influenza results and the potential and the directions that we can go from here. So for me, in my mind, this is a short outbreak. COVID is going to be a short outbreak, should not change anything. And I resisted that for maybe, at least for the second half of February. For the whole February, I was living in denial, basically. I think inside and deep in myself, I knew that this is not going away like what I think it is. But I was trying to convince myself. And at that time, I was really consumed to finishing our paper and submit it. And once the paper was submitted, I felt more relieved. But then we started quickly seeing the scale, and this is not going to go away easily. So the advantages, and we can discuss that, is really what I always say. I was a bit disappointed by the shutdown and our termination of all our flu studies, and everything has to be discontinued. But eventually, I was very disappointed for some time, but then I started to realize the opportunities that, at least scientific, but also, scientific opportunities that COVID brought for us, and what kind of new studies we can do.
Further up on that, so since you were set up to essentially look at this draining lymph node, how have you used that now to look at infection or vaccination in the pandemic with COVID?
Yeah, so that's what we have. That's exactly what helped us, that we were basically set for a vaccination setup. So infection, the draining lymph nodes are, especially for respiratory pathogens, these are in the peribronchial lymph nodes, or basically they are in the lung, and it's hard for us to access. I definitely inquired about that, and we basically, I was told it is doable. There's some degree of risk in it.
Yeah.
That I'm not sure was really worth it at that time, or the questions weren't that degree of risk that to sample using the same lymph node, ultrasound-guided lymph node aspiration with needle, to actually sample these nodes in the lung during a bronchoscopy procedure. So I thought, okay, no, infection was hard. So we did the same. We looked like everyone else. We looked at the immune response in blood. In patients, we were actually behind in Missouri, so we didn't get really patient samples starting in April, although other places, on the coast, New York and Washington State, started in February. So we were a bit behind in that. So there are already some data coming out at that time. We are starting to get our samples. But when the vaccine became available, we really had the setup, because it was giving intramuscular in the upper arm, same as in flu. So we had the whole setup really ready to go. And the whole team was very basically poised to jump on it. And we did actually did very um, we started asking again the same important question, simple ones that we have. What kind of how robust the response is? Everyone can look in blood and look at the serum antibody titers, but what's happening in the germinal center? And actually, the influenza setup helped us in many ways, because now we actually can appreciate influenza act here as a control, because we can see how with a very mild immunization like influenza, what's the difference. It provides like almost a scale for how robust the response after this, at least the mRNA-based vaccines, that the ones that are available now. So the reaction was really robust. I remember Sherry really mentioning that the lymph nodes are really different. And when I tried to ask how different, she said, they are just very big and juicy. And that was the expression. They are just really, and then you start reading reports in CNN and other news outlets that this is really, some people are actually can feel the lymph nodes. And they said, well, great, this makes sense. They are great. They are big.
Yeah. Yeah, that actually happened to me. Yeah, I didn't have any of on the second shot, I didn't have any other symptoms, but what I actually felt was twinging of the lymph nodes in my armpit. I've never actually felt that for a vaccine before.
Yeah, so in that case, we probably didn't need ultrasound, but we did use ultrasound still, and we did this in our healthcare workers. We did our first study, and this is under revision now. And we looked at you can tell the germinal centers are just massively expanding in those lymph nodes. And then we see something that we didn't even see. We see like multiple nodes involved. In flu, if we get one node, we are really excited, very happy. And if we get one node for one time point, we are very, very excited. For here, it's like we are like spoiled by riches, like just with so much responses and multiple lymph nodes, and to the extent that you think to the extent that I want them to calm down so I can actually now focus on finishing up the story instead of having this open-ended. When this actually till now after vaccination, people are vaccinated in December, and germinal center is still ongoing. So I'm not sure how long it will be going. It's exciting. Just at a certain point you think, okay, let's wrap it up.
So I guess, what does that mean if you have such an active germinal cell response for like the quantity of the B cell response or T cell response, or the quality in a way of that response? What does it mean to have a very active germinal cell response?
So that's a great question. So I guess we can go from the beginning. As we know that if germinal centers are really important for not only making which is the most famous part, is introducing mutations in the B cells, or where B cells acquire mutations to actually refine their binding. Basically, we are making the cells better binders. And this is through a really a competition almost between the cells and each other. And the ones the best binder ones usually win. At least that's the dogma at the moment. And those are the ones that we will continue with for the rest of our lives as memory cells, as cells that long-lived cells that secrete antibodies for us. So that's what happens when we get our childhood vaccines. We get those vaccines, and for the most part, we never need any booster immunization for those. And those, because we have a beautiful immunization at that time that most likely induced a very robust germinal center that induced these memory cells and memory plasma cells, memory B cells, memory plasma cells, that secreted all, that kept us, safe till now. So a very robust germinal center means that you have a bigger chance to induce more of those memory cells. and you have a longer operation of actually refining the process of refining which cells can the cells that this competition is basically becomes more protracted, and that means that the cells will again theoretically will accumulate more mutations, and that accumulation allows us to be even higher or stronger binding to the vaccine and to the virus. So becomes the antibodies secreted by these cells are even better protector, or can mediate better or enhance protection. So and then you get more of these cells in our uh, pool of memory. So we are in a way, it's so far there's no disadvantage. There's a lot of good things that coming from this reaction continuing. And I think one of the things that we know that adjuvants, which are the materials that we add to vaccines to improve their immunogenicity and to make them more potent, are actually one of the most important part they do is they actually enhance the germinal center reaction. And many adjuvants are that's how they improve vaccines. So now we have a vaccine that actually almost self-adjuvanted that can really drive that. So that could be only can be good news for durability of the response and for how mature is the response. And that's something obviously we should be able to tell in the next few months.
And what does this mean for when you have to boost or something like that? So I know people are starting to, chatter a little bit about anagenic sin, and if we have to then come in with booster vaccines that basically are covering new variants or what have you. What does it mean if we have like really good germinal centers when we actually have sort of this another viral vaccine against another viral strain?
Yeah, so that's the a really where the flu vaccinologist, immunologist, and influenza people has been in really immersed for the last probably starting from the late 40s or actually early 40s when they realized. So the virus was first isolated in 1933, and then we thought we have a vaccine in the late 30s, and the vaccine was working great. It was based on formalin activation of eggs of the virus that was grown in eggs, so it wasn't the cleanest, but it worked at the time. But then when the first variants, or not that case, drifted strains started to show up, then we realized that we have a problem. And it took us 10 years at the time to realize the magnitude of the problem, that that previous immunity that induced by this exposure might not be very helpful. At the time, Tony Francis, who was an epidemiologist, very insightful epidemiologist at the time, came up with looking at the responses to infection across the multiple individuals over time, but across different age groups, and linking that to what was the most, at the time there were only few viruses that were really characterized. So he linked that to what are the first virus that those individuals likely exposed to, and he came up with a really very amazing way. And I guess it's based on his religious upbringing. He brought us the term original anagenic sin, just because you always respond the point is you always respond to what you have been primed with or conditioned with or imprinted with in your childhood more strongly than what you respond to the more recent strains. And in a way, he considered it a sin. At the time, of course, again, he used only serology to really come up with the term. The concept of immune memory wasn't, the clonal selection theory wasn't very established, and it wasn't actually even known when he came up with these terms. So it was just at the time lots of very preliminary proposal about the cellular component of the immune response.
Right.
So now he comes with that, and we know that this is now immune memory. And so going back or going forward for 80 years, and we are still, I think, for influenza, we're still struggling with this point, is how we can immunize a partially immune population. Because if you all of our most successful childhood vaccines, or vaccines in general, even the, the vaccines like yellow fever and others that are given to adults, those vaccines are given to naive hosts, and those are the most successful. When the challenge for us has been to give vaccine like influenza to and similar story like in malaria. The challenge was always when you give it to people who are actually partially immune. The story with malaria vaccines being more works better if you try it here in the US, but if you take it to the endemic population or endemic areas, they don't do as well. In terms of immunogenicity, I think it's clear that it's this pre-existing immunity are actually interfering with and that was actually the topic that we were really trying to understand by looking at cellular immune responses to influenza. Now with COVID, with SARS-CoV-2, I should say, that it's still unclear. We definitely some of the variants of concern really can have mutations that are, non-scientific term, annoying in terms of they are mutations in places where neutralizing antibodies, protein ones, are targeting. But I don't think we are at the point yet where we can decide that we really need to update the vaccine.
Right.
And if we, is it really, it seems that the vaccine, even if we lose some of the neutralizing capacity, there are lots of other antibody responses and other non-B cell responses,
right,
T cell responses that could provide that edge of protection. So we could potentially be going for that direction with these viruses, that they will mutate, and they will change enough for us now that we have to update the vaccine. And for that, we now our studies will be the important question will emerge from that is what happens when you immunize with a novel strain or in that case a variant from SARS-CoV-2. And now the situation will be different because we will have memory cells that have targeted that were again induced by a robust germinal center reaction against the first circulating strain, the Washington strain that most of the vaccines are based on. So in that case, now it can the new immunization induce another robust germinal center that can recruit naive flows that are only specific to those epitopes that are, or the determinants and the new variants. And I think that based on our flu work, which is, again, that was the work that it seemed that the germinal center has that capacity to actually engage not only the memory B cells that have been generated in the previous years, but also in a smaller fraction of cells, also naive B cells can be engaged. And I think from that, if I extrapolate from that, I think that could be the case also with SARS-CoV-2.
And does that sort of reflect on maybe why people that were previously infected with and had COVID, are now, when they actually have sort of like a boost with the vaccine, we're still seeing good immune responses? So I think, there was some fear right at the beginning that potentially if you were naturally infected, you would actually not have good immunity, as it were, and that might even poison, as it were, the vaccination later on. But that does, at least in the quantity of the response, that doesn't seem to be happening. I don't know. Are you going to do studies on the quality of the response after natural, then vaccination, natural infection, vaccination?
Yeah, so that actually happened by chance in our study that I mentioned, because the people, the first few subjects or participants that we recruited, some of them already had a pre-confirmed, previously confirmed infection.
Oh.
And so we didn't really design it this way, but those are healthcare workers, so they were exposed to the early wave. And they were also recommended to get the vaccine like everyone else.
Right.
And then it was very clear when you look at their response to the first dose that these people had experience. And obviously they've had mounted, and many studies have shown that now they mounted a great to the first immunization, both quantitatively and qualitatively. Actually, it is a
Okay.
broad response. There is really no reason to think that, and again, but that was most likely a homologous
Right.
boost. So it's a different part, because we are giving them the same engine, that in that case it's giving them in a different form.
Right.
And infection remains the gold standard for any immune induction. Really, it's mild virus infection is probably more immunogenic than, and better immunogenic than any vaccine.
Right.
And most successful vaccines are actually just live attenuated viruses, right?
Right. Okay, well, cool. That was very interesting. And just to talk a little bit generally as we're finishing up, I mean, how has the, working on this, but also personally, how have you fared in the past year? How has the pandemic affected you? And then thinking about vaccination and things like that, how do you sort of deal with or talk about vaccination with your friends and your family?
Yeah, we can spend another episode about this. Yes, that's I don't know where to start from. So I have three children who are in elementary school level, young elementary school level. So that has by far was extremely challenging. By all measure, I never thought. You would think that having a 6, 7, or 8 year old sit on an iPad for that would be like their dream, which probably their dream. I don't think that was meant. I think it in their mind this was an entertainment.
Yeah, not school.
Yeah, not really. It's not associated with knowledge or getting anywhere. So it was a challenge from that part to that was the first part, especially in the starting this, in the first shock we had, like April and last year wasn't really bad. It just August, September, and October and November. This last fall semester, this one was really brutal in terms of really adjusting learning and adjusting, and the changing between going to school and staying at home and trying to adjust our lives. So that's basically, and who is going to school, who's staying at home, and if those who for the ones that are going to school, who's going to pick them up, and who's that, and they are working full time, and then the call that someone tested positive, everyone stays home, and teacher tested positive, and all of, yeah, we went through a lot. I think not only us, I'm sure we were all, I mean, everyone in the same stage went through a lot. I mean, I have to be, and I'll be frank, I mean, being that I have flexibility at work helped me, and helped our family, because I don't have to be, thankfully, I'm not doing the experiments with my hands, and thankfully, but then I can only imagine how hard this was for postdocs or graduate students who had families and had to go through this. This has been, I'm just happy I'm not a postdoc at that stage. I mean, I can only imagine. It just has been really hard.
Yeah. And do you still have family back in Egypt? How was that part of it?
Oh, this is you're getting to the yeah. This is the part that we are still living through. Right now, actually, it's really bad.
Yeah.
And I do still have family in Egypt, and I call them every day, because it's just yeah, it is very bad, because you realize right away when you live in, I've been living here for 15 years now, but you realize right away social distancing and many of the measures, working from home and all of that, it's almost a luxury for many places, and definitely a luxury for developing countries, and that has been hard to, when you are, when your livelihood is actually basically means that you go every day outside in the street, and and giving it the population density in Cairo, for example, it just that social distancing, the concept itself is not even there, and then add to that that masking is just not in the culture, and 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 again, it's still a luxury, and still the case up till now. It's amazing that lots of people that it's amazing when you see that even if I've been away for 15 years, I've known so many people in the last few months that passed away that I've known them personally. I've met them.
Wow. Yeah.
I spent time with them. Some of them are relatives.
Yeah.
Some are acquaintances, and some are many friends have been infected in this. And just one of those things that are, you know, making me concerned every day. That and unfortunately, it's one of those things that traveling is not an easy. It's doable. It's not easy. And you wish if you can do something.
Yeah.
And that's the hardest part. That you basically cannot do something. And you can only, and it makes you just wonder when you see people here not getting the vaccine, although they have the option. It's very easy for them to get.
Yeah.
Is I wish if I can take some of this vaccine and give it to people that actually would stand in lines for it, but there's not really a vaccine there. And just that's a part that we have. Probably every immigrant from many places, you hear the same stories from people coming from India, from Brazil. But it's all the concern at the moment about home, and hoping that at least the wave that they are going through right now, that vaccination so far have spared us here in the US.
Yeah.
That this wave will go and without really much more damage than it already did.
Right. Well, thank you very much. It was great talking to you. I'm always interested in your research, and it's really cool to see how you were able to, as you say, sort of pivot into the SARS research using this almost a established platform that was very timely, right before the pandemic started. So thank you very much.
Thank you for having me. It was absolute pleasure. Thank you.
Ollie's work using samples obtained from ultrasound-guided fine needle aspiration of draining lymph nodes of human volunteers following their mRNA vaccination against SARS-CoV-2 shows that this vaccination drives a robust germinal center reaction and promotes a durable and mature humoral response to SARS-CoV-2. This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcasts, or at lmtv.podbean.com.